Welcome to our in-depth module on quality control. In this video, we'll explore critical aspects of quality control in the pharmaceutical industry, including chemical and physical analysis, instrumental techniques, and good laboratory practices. We'll also discuss standards, microbiological applications, impurity analysis, calibration, packing material analysis, and post-release monitoring. Let's dive in. Quality control begins with thorough chemical and physical analysis to ensure that pharmaceutical products meet required specifications. Chemical analysis. The objective is to determine the chemical composition and concentration of active ingredients and excipients in a drug product. Techniques include titrations, spectroscopy, and chromatography. Physical analysis. The objective here is to assess physical properties such as particle size, bulk density, and solubility. Techniques include methods like microscopy, sieve analysis, and rheology. Instrumental analysis provides precise and accurate measurements critical for quality control. Let's look at some common techniques. Gas chromatography, or GC. Its purpose is to separate and analyze volatile compounds. It's commonly used for purity testing and quantifying active ingredients. High-performance liquid chromatography, or HPLC. This technique is used to separate, identify, and quantify components in a liquid sample. It's widely used for analyzing complex mixtures, including drug formulations. Ultraviolet spectroscopy, or UV. This method measures the absorbance of UV light by a substance. It's useful for determining the concentration of compounds that absorb UV light. High-performance thin-layer chromatography, or HPTLC. This technique separates components based on their interaction with the stationary phase. It's often used for qualitative analysis and purity testing. Standards are essential for ensuring the accuracy and reliability of analytical results. Reference standards, or RS. These are pure substances used as a benchmark for the analysis of pharmaceutical products. They ensure that test methods are accurate and reproducible. Working reference standards, or WRS. These standards are prepared from RS and used to calibrate instruments and validate analytical methods. They verify the performance of analytical methods during routine testing. Good laboratory practices, or GLP, are crucial for ensuring the reliability and reproducibility of laboratory results. Principles of GLP include organization and personnel. Ensure that laboratories have qualified staff and defined roles. Equipment and facilities. Maintain and calibrate equipment and ensure a controlled environment. Documentation. Keep detailed records of experiments, procedures, and results. Quality assurance. Implement procedures to monitor and assess compliance with GLP standards. The benefits of GLP are numerous. Consistency. It provides reliable data for regulatory submissions and decision-making. Compliance. It ensures adherence to regulatory requirements and industry standards. Microbiology plays a vital role in ensuring the safety and efficacy of pharmaceutical products. Microbial testing has several key objectives. Objective: to detect and quantify microorganisms in pharmaceutical products. Techniques: this includes tests for total microbial count, specific pathogens, and sterility. The application of these tests is crucial. Sterility testing ensures that products intended for sterile use are free from viable microorganisms. Microbial limit tests measures the levels of microbial contamination in non-sterile products. Understanding and controlling impurities is crucial for product safety and efficacy. The International Council for Harmonization, or ICH, guidelines provide a framework for impurity testing. Known impurities. Definition. These are impurities identified during the development phase. Control. It's important to monitor and control these impurities to ensure they are within acceptable limits. Unknown impurities. Definition. These are impurities not previously identified or characterized. Control. Use advanced analytical techniques to detect and quantify unknown impurities and assess their impact on product quality. ICH Guidelines. Guidelines. These provide detailed recommendations for impurity testing and control, including limits for various types of impurities. Calibration is essential for ensuring that analytical instruments provide accurate and reliable results. So, let's break it down a bit. Purpose. Accuracy. Verify that instruments measure correctly and produce reliable data. Maintenance. Ensure that instruments remain within specified performance limits. Procedure. Standardization. Use certified reference materials to calibrate instruments. Documentation. Record calibration procedures, results, and any corrective actions taken. Frequency. Routine calibration. Perform regular calibration as part of a maintenance schedule. Verification. Conduct additional calibrations as needed based on instrument performance and usage. Packing material analysis ensures that packaging components are suitable for protecting and preserving pharmaceutical products. Analysis. Material integrity. Test for physical properties, such as strength, flexibility, and barrier properties. Compatibility. Assess the interaction between the packaging material and the drug product. Purpose. Protection. Ensure that packaging materials maintain product stability and prevent contamination. Compliance. Verify that materials meet regulatory requirements and industry standards. Deviations from established procedures can impact product quality and regulatory compliance. Planned deviations. Definition: pre-approved deviations from standard procedures, often used for validation studies or process improvements. Documentation. Clearly document the rationale and approval for planned deviations. Unplanned deviations. Definition: unexpected deviations that occur during manufacturing or testing. Investigation. Conduct a thorough investigation to determine the cause and impact of the deviation. Corrective actions. Implement corrective and preventive actions to address the deviation and prevent recurrence. Post-release monitoring is critical for maintaining product quality and addressing any issues that arise after market launch. Complaints. Objective monitor and investigate complaints related to product quality or adverse effects. Action. Evaluate complaints, take corrective actions, and communicate with regulatory authorities if necessary. Recalls. Objective. Remove defective or unsafe products from the market to protect public health. Procedure. Follow established procedures for initiating and managing recalls, including notifying stakeholders and regulatory agencies. Post-market surveillance, ongoing monitoring. Continuously track product performance and safety through adverse event reporting and market feedback. In this module, we've covered key aspects of quality control, including chemical and physical analysis, instrumental techniques, good laboratory practices, and more. Understanding these elements is crucial for ensuring the safety, efficacy, and quality of pharmaceutical products. Thank you for joining us, and we look forward to seeing you in our next module. See you next time.